Good afternoon. I'm Serge Belanger, the Spec Pharma analyst here at Needham, and I'm proud to have Acer Therapeutics with us this afternoon, and we have the CEO, Chris Schelling, who's here to tell the story of Acer.
Thanks, Serge. So I'm Chris Schelling. I'm the CEO and the founder of Acer Therapeutics. Acer is a pharmaceutical company that acquires, develops, and we seek to commercialize therapies for serious, rare, and life-threatening diseases with critical unmet medical needs. We're headquartered up in the Boston area. We have about 40 people in the company. We were founded a little over 5 years ago and went public via a reverse merger with Opexa Therapeutics in September of 2017. At the end of last year, we reported cash of $41.7 million and are expected to have sufficient capital into first half of 2020. The team has grown very quickly recently, and we have a very established, experienced leadership team that has both extensive orphan drug development experience as well as launch experience. So I think we've assembled a really nice team that's well poised to take our products through development and potentially into commercialization as well. A quick overview of the company. We're going to get into additional details on the following slides, but on slide 5, the pipeline at Acer consists of 3 clinical stage product candidates. Edsavo is our lead compound. This is for the treatment of vascular Ehlers-Danlos syndrome with a type 3 procollagen mutation, a COL3A1 mutation. We have a second compound. This is a pre-NDA compound. Acer-001 is a fully taste mass and immediate release formulation of FDA EMA approved product called sodium phenylbutyrate that we're developing for various inborn errors of metabolism, including urea cycle disorders, maple syrup urine disease, and most recently in January, we acquired worldwide rights to a compound called Esanetant from Sanofi. We have not disclosed the indications. We'll talk a little bit more about the target, but we're going to be looking at a variety of different neuroendocrine disorders to take that program into the clinic. So our candidates really have a comparatively de-risked profile, having one or more of the following profiles, either a favorable safety profile, clinical proof of concept data, mechanistic differentiation, and an accelerated development, a regulatory development pathway, including priority review or 505(b)(2), for example. And we've had multiple milestones recently and coming up that are of critical importance. At Edsavo, we actually had the NDA accepted with priority review in December 24th of last year, which puts our PDUFA action date on June 25th of this year. We anticipate filing the IND for Esanetant in the second half of this year and a potential NDA submission sometime in the first half of 2020. As I mentioned before, we should have enough capital to take us into the first half of 2020. Slide 6 is just a graphical representation of the pipeline, and 7 is a summary of our lead compound, Esanetant. I'm actually going to be getting into additional details on the subsequent slides. So on slide 8, so Edsavo is to date, it's the only agent that's demonstrated clinical benefit in vascular Ehlers-Danlos syndrome. The compound was originally approved back in the 1980s in Europe for the treatment of hypertension. At the time, the compound was known as a beta-1 antagonist. As the drug was further studied over the decades in Europe, it was determined to have a number of different mechanisms of action, and thus the investigators that have thoroughly studied this drug have concluded that it has a very unique pharmacologic profile, including beta-2 and beta-3 agonism, alpha-2 antagonism, and the drug, independent of the beta-adrenergic receptors, upregulates endothelial nitric oxide synthase. So you can see on the right-hand side of slide 8 how this compares. So a lot of people will say, oh, this is a beta blocker, so how does that compare against other beta blockers? In fact, In the VEDS population, the disease that we're specifically looking at getting this drug approved for, the beta-1 antagonism is not a relevant indication. In fact, it's the other mechanisms, the beta-2 and beta-3 agonism in particular, that lead to vasodilation, smooth muscle relaxation within the vasculature that extend the benefit, the treatment benefit in this patient population. In fact, most VEDS patients are normotensive, they're younger, and so, the properties of a beta-1 antagonist in lowering blood pressure or whatnot, really, you don't see that kind of benefit in this patient population. So, on slide 9, a little bit about the disease that might help to further characterize this. Vascular Ehlers-Danlos syndrome is a subtype of a broader spectrum of disorders called Ehlers-Danlos syndrome. The thing that's in common with all of these disorders are that it's a defect of collagen synthesis, and so different genes encode different types of collagen throughout your body in different tissues. So the classic form of the disease means that you don't produce collagen in your skin and you have very stretchy skin. Other forms, the hyper-extendability or hypermobility form, it means you don't produce collagen in your joints, and therefore you become kind of a contortionists have this issue. They have dislocations of their joints. But in the vascular form, which is specific to type 3 procollagen or the COL3A1 gene, that encodes collagen that's found in blood vessels and hollow organs like the intestines, the uterus, etc. And as such, whereas the other diseases are interesting in the sense that you have stretchy joints and skin, in this disease, it's characterized by aneurysms, dissections, and ruptures of the vasculature, the gastrointestinal and uterine tissues. And as you can imagine, if you have very weak tissues in those areas, you have significant morbidity and significant mortality as well. The gene defect is autosomal dominant, meaning 50% of patients who have the disease have inherited it from their parents. And so we do know family members that, there's 5 or 10 family members that have the disease. The disease is also spontaneous, which means you don't have to have a family member who has a mutation. You can just inherit, you can just be born with a mutation in COL3A1. The disease itself is typically diagnosed in your teenage years. About 25% of patients are diagnosed before the age of 20. Nearly everyone is diagnosed or has a clinical event by the age of 40. And it's diagnosed through clinical symptoms, meaning one of these patients may have a colon rupture or a vasculature rupture or dissection that leads them to the ER. They're young, and so the clinician tends to go provide genetic testing to determine what's going on, and they come back positive for COL3A1, and that's how these patients find out that they're positive for VEDS. The median age of death in these patients is 51, so in the United States it's closer to 80. So you can see a significant fall off in survival in this patient population. And there are no effective therapeutics for these patients with VEDS. Current treatment is focused on surgical intervention. Once you've had an event, you go into the ER, they repair it via surgery, and you wait for your next event to occur. And you can see on the right-hand side of slide 9 that there's no predisposition for where an event may occur. When we're just talking about the vasculature, it can happen in the cerebral area, it can happen in your aorta, it can happen, in all different parts. And a lot of these patients will have events that just happen around the body. And you can have a gastrointestinal rupture and then an aortic dissection. There's no rhyme or reason for it. So these patients, you can imagine, they're ticking time bombs, and they know it, and they're just constantly waiting for the next event to occur with no therapy available to them. Which is why we're introducing IDSIVO here in the United States, potentially if it's approved. The pivotal clinical trial that supports the data was run in France. It was a randomized trial conducted at 9 centers throughout France and Belgium. The study enrolled patients from 2002 to 2009 and published data in 2010. And you can see this data was published in 2010 in Lancet. The primary endpoint was time to first event or event-free survival analysis. The trial was stopped early for clinical benefit. It was meant to go for 5 years. It was stopped at 4 years of follow-up. The primary endpoint were vascular events, either dissections and ruptures, and you can see in the upper right-hand corner of slide 10 that by the 4th year of follow-up, there was a statistically significant difference in terms of time to event by being on therapy versus not being on therapy, with a hazard ratio of 0.36, meaning the relative reduction in your risk of having an event by being on drug was 64%. When you factor in other clinically relevant events, these were gastrointestinal and uterine events, that's what you see down on the right-hand side was your time to first any clinically relevant event, and you can see the p-value dropped to below 0.01, and the hazard ratio dropped to 0.31, or a 69% reduction by being on drug for all clinically relevant events. Very, very meaningful data. And the author's comments at the time that this was published was that they suggested the drug would in fact be the treatment of choice for VEDS patients throughout Europe, and it has become the gold standard over the last decade or so throughout Europe for VEDS patients. Once the trial was completed in France, they enrolled all VEDS patients into a registry, so there were a little over 400 patients throughout the country. The one center in Paris is the largest academic hospital system. They enrolled 144 patients in this registry. All of these patients were positive for the COL3A1 genetic mutation, and over 80% of these patients were on celiprolol. This was a very robust registry in that they followed patients prospectively on drug for a number of years, a median follow-up of about 5.5 years, and they collected retrospective data on these patients such that every patient in this registry had up to 17 years worth of data follow-up. So this is pending publication in a top-tier journal. We believe that this publication is imminent, and we're excited to see it, but we think that the data that will be published is looking not at time to event analysis as what happened in the randomized trial, but rather looking at mortality, overall survival, as well as rate of hospitalization in these patients. So, and that was on an intra-patient basis. So if a person was put into the study, the time that they were on retrospectively that was collected before they were on drug was compared to the time that they were on celiprolol, and was determined whether there was any difference in the rate of hospitalization by being on therapy. So we're excited to see this data get published pretty soon. Moving from development into commercialization, since PDUFA is coming up at the end of June, the launch plan is pretty straightforward and it is common in all ultra-orphan compounds. So the launch plan is really 3 pillars. It's patient identification, establishing a network of excellence in the United States that has a special focus on VEDS, as well as market access ecosystem. So in patient identification, the goal is to identify, we know that there's about 2,000 patients that are currently COL3A1 in the U.S. We believe there may be as many as 5,000 patients with the disease and the genetic mutation. Our goal is to go out and provide genetic testing for those patients to bridge that gap, and so this is an effort that's currently underway with our commercial team. The network of excellence, the goal here is that a lot of these patients don't have a current provider who is specialized in VEDS. A lot of them are managed by geneticists in the United States who, because there's no therapeutic available to treat these patients, they kind of manage and oversee them. But what we'd like to do is move them maybe more into vascular centers that can do imaging and prescribe the drug once it's available. And so we're establishing 50 centers at launch that really specialize in the vascular care of these patients and can prescribe the drug, and also have an opportunity to participate in evidence generation strategy, additional publications once the drug is available. And lastly, when we talk about market access ecosystem, we want to ensure that this drug has optimal pricing and access for all patients who are COL3A1 positive. In fact, we've prospectively asked that COL3A1 positivity be included on the label for the drug. And we want to make sure that there's robust patient and physician support services so everyone has the availability. If you're COL3A1 positive, we want you to be on this drug. Slide 13 is a quick overview of the commercial and medical affairs leadership. As you can see, again, extensive orphan and launch experience on this team. We've already hired 6 sales reps that have begun reaching out in the field, and a number of MSLs as well. We'll be doubling the field presence here as we get closer to launch, but we really have a nice team here that is capable of launching this drug successfully into the U.S. A little bit more about the market sizing. So I alluded to 2,000 patients earlier, potentially as many as 5,000 patients. The way we know that there's 2,000 is we actually surveyed the labs that provide COL3A1 genetic testing, and through that survey, we're able to determine that there were about 2,000 patients in the U.S. that have been currently identified. We wanted to find out, because we've talked with a lot of families, and they've said, we have family members who have not been tested, so we were wondering if there was a way for us to be able to identify them and pick them up and see how many patients might be in the U.S. with VEDS. And so we looked at payer databases. We thought this would be a good place for us to start. So we hired a group called HVH that looked at the Truven Market Scan database. This is a database that has about 190 million covered lives in it. And through that effort, we identified EDS patients who had an ICD-9 or 10 code, and then a comorbid vascular component of the disease. And through that effort, we were able to identify 4,200 phenotypic VEDS patients out of that 190 million lives. And that extrapolates to roughly 7,000 patients when we think of, over 300 million people in the United States, the general population. Now, not all of those folks are going to have a COL3A1 mutation. That's our effort to go out and find out how many patients are. So our conservative estimation is somewhere between 2,000 and 5,000 patients in the U.S. with a COL3A1 mutation. The way that we want to bridge this is, in fact, by providing a broad genetic testing program for all patients. And so we're currently working with a genetic testing program. This would be a commercially available kit. This would be a saliva-based test that we would provide. So once a known COL3A1 patient in a family is identified, we want to go out to all of their family members. Since this is autosomal dominant and they have a 50% chance of having the disease, the goal would be to provide broad genetic testing so that we could identify a few additional family members within that family tree who might, in fact, be positive for COL3A1. And this test will be rolling out here in the next couple of months. This will be free of charge to patients. We work very closely with patient advocacy. In fact, this last Saturday, we had a patient meeting, had over 35 patients and family members attending an advisory panel in Washington, D.C. So we have a very close relationship with patient advocacy, social media, and whatnot in this area. This is all in the effort to identify additional patients and work closely with them and their family members to keep engaged with them. And as I alluded to earlier, in terms of establishing the network of excellence, the goal is to have 50 centers by the time of launch. The focus of this would be in vascular medicine, but in fact, because of the nature of the disease, there's a lot of subspecialists that we want to engage as well, including neurology, high-risk obstetricians for women who are pregnant, geneticists, emergency medicine surgeons, etc. So this is kind of an extensive effort. Obviously, it just takes one physician to prescribe, but we want coordinated care. The goal is not just to provide a drug for these patients, but also to provide all of the support that's necessary to ensure optimal outcomes for these patients. Lastly, as we mentioned, and this is a very busy slide on slide 18, it's a little bit about market access. What I can say is we're not recreating the wheel. We've done extensive research with payers and PBMs to determine what would be the optimal price and what would be the optimal support for these patients. And Acer is committed to establishing care programs that ensure if you have COL3A1 disease, you'll have access to the therapy. What form and fashion that takes, whether that's patients who are insured and we provide support programs, or whether that's a free drug program, we're looking at the full range of support for patients to ensure everyone has the availability to get onto drug. And lastly, I'll point out that because of the richness of the data that's in the payer system, in particular for patients in the U.S. for which this drug's not available, we're able to generate some significant health economic and outcome research to understand what the current cost of care is in the United States for these patients who don't have a therapy. And so we'll be looking to publish this HEOR data later this year that talks a little bit more about what current cost in the event that they have an acute event, what that cost of care is. And I can tell you it's quite expensive. So there may be some cost offset relative to introduction of a therapy here in the United States in the event that the drug is able to prevent these acute events from occurring and hospitalization that goes with that. So in summary, for Edseva, if it's approved, the drug becomes the only FDA-approved therapy to treat these COL3A1 VEDS patients. There are up to 5,000 COL3A1 VEDS patients in the United States, and our focus in the field is in those 50 centers at launch, potentially expanding that to around 100 centers within 3 years. We know that orphan pricing is well supported by the initial payer research, with additional validation from the pharmacoeconomic studies that we'll be publishing later this year. And again, we'll be making sure that everyone has access to this drug through robust patient assistance programs, et cetera. And a quick note that the drug has been granted orphan drug designation. That's 7 years of market exclusivity. We would be seeking an additional half a year of pediatric exclusivity, so we're looking at roughly 7.5 years of marketing exclusivity for the compound. And we have filed a number of use patents at the end of last year that, if those issue and get put into the Orange Book, could extend the runway beyond that 7.5 years. Moving on to our second compound, ACER-001. Again, slide 21 is a summary of the compound. But in a nutshell, this is a drug that is FDA and EMA approved for the treatment of urea cycle disorders. These patients have defects in 6 enzymes of the urea cycle, which is found in the liver. This is the enzyme system that removes ammonia from patients. And what's been known is that phenylbutyrate is a prodrug of phenylacetate and PAGN, and those are in fact the active moiety in removing the nitrogen, the ammonia from these patients. There are a number of treatment options for these patients. There's a little over 2,000 urea cycle patients in the U.S. About 600 of them are currently on a formulation of sodium phenylbutyrate. We'll also be studying this drug in another disease called maple syrup urine disease. This is an inborn error of branched-chain amino acid metabolism, very similar to PKU, except that it's in 3 of the 9 essential amino acids, leucine, isoleucine, and valine. The enzyme that is affected in here is called the BCKD complex. Unlike in urea cycle, it's not found in the liver. This is found in skeletal muscle and mitochondria. And this enzyme is defective in these MSUD patients. There's about 800 patients in the U.S., 3,000 worldwide with MSUD. And you can see on the right that these patients are severely affected. They tend to have lower IQ. They have cognitive and social impact on the disease, and in acute settings where leucine in particular gets to very high levels, these patients can go into a coma and die. No therapy available to these patients other than a highly restricted diet where 90-95% of their diet is synthetic food and formula, and they have to weigh out fruits and vegetables daily. So huge unmet medical need here as well. What's interesting about MSUD is that it's a different mechanism of action, and the reason why phenylbutyrate was determined to work in MSUD was that in the vast experience of treating urea cycle patients, they noticed an off-target effect in MSUD patients. You can see that in the upper right-hand corner. There's no reason why giving phenylbutyrate should lower branched-chain amino acids, and yet you can see in over 550 patients, that's exactly what physicians were seeing. And so Dr. Brennan Lee and his colleagues at Baylor College of Medicine further explored this, and they found that the drug has a novel mechanism of action in MSUD. So in urea cycle, phenylbutyrate gets converted to phenylacetate that removes the ammonia, and in MSUD, phenylbutyrate is in fact the active moiety, and it inhibits a kinase that regulates the BCKD complex, thus leaving it constitutively activated. And you can see on the lower right-hand side, when they tested this, not only in healthy subjects but also MSUD patients, you can see that all of the healthy subjects had a lower branched-chain amino acids levels, and MSUD subjects, 3 of the 5 patients that were studied had lower BCAA levels as well. So being an allosteric inhibitor, the drug basically hypermetabolizes all branched-chain amino acids, and that's clinically relevant in the MSUD population. So the compound ACER-001 itself is in fact an immediate release taste mask formulation. You can see on the left-hand side it's a pH-sensitive polymer that coats the phenylbutyrate, and in a neutral pH the drug does not dissolve, but in an acidic environment like the stomach, the polymer dissolves completely within about 15 minutes, so that by the time it reaches the intestines, it's bioequivalent to the original drug Bufenil. Now, quick background on the forms of phenylbutyrate that are available. There are 2 drugs that are currently available. Bufenil, that's the immediate release non-taste mask drug, and then there's Ravicti, which launched in 2013. This is a prodrug, a slow release formulation that is taste masked. Bufenil right now is around $150,000+ a year. That's the generic. And then you have Ravicti, which is now a little approaching $900,000 per patient per year. So when you look at it, the potential for these 3 compounds side by side in the upper right-hand corner, you can see all 3 drugs are going to work in urea cycle. They're going to be efficacious and safe. In MSUD, because phenylbutyrate is the active moiety, you need an immediate release formulation, you need a big Cmax of phenylbutyrate, and that's only Buphanyl and ACER-001 are expected to be efficacious in MSUD. When we talk about palatability and compliance, that would be the taste mask formulations, Ravicti and ACER-001. And I'll point out that Buphanyl, the original drug, is butyric acid. It's a form of butyric acid which is found in vomit, in rancid butter. And these patients take up to 20 grams a day. This is not a fun drug to take, so taste masking is critical in this patient population. It's not to be diminished. People who were on Buphanyl originally, their compliance was less than 40%, so obviously they had suboptimal outcomes. But when we talk about reasonable orphan pricing, Ravicti is getting up to close to $1 million a year. That's really egregious pricing. So our goal is to provide a reasonably priced therapy that is taste masked for both urea cycle and MSUD patient populations. Once we launch, the goal is to do a bridging study, a PK study, to show bioequivalence to Buphanyl in the second half of this year. And our job would be to target the existing phenylbutyrate market share within urea cycle. We're not trying to add patients. We're literally trying to cannibalize patients who are on either Ravicti or Buphanyl and move them over to ACER-001. You can see here revenue for those drugs in the United States last year was almost $250 million. We will do this by a well-differentiated formulation and competitive pricing. We'll then expand it into MSUD we'll do phase 2, phase 3 studies in MSUD. And we have filed patents on our formulation. We have issued use patents in MSUD. We'll have some market exclusivities afforded to this drug as well, and seek additional pediatric work to extend a pediatric exclusivity. And like with Odsivo, we'll be providing robust support to these patients as well. Lastly is our drug we just licensed in January, Asanitant from Sanofi. We got worldwide rights to this compound. We have not disclosed the indications of interest, but we can talk a little bit about the neurokinin 3 receptor for which Asanitant was developed. So this compound is considered the reference drug of NK3R antagonists. It was originally developed by Sanofi in the 1990s and 2000s for schizophrenia. It was put in over 1,000 patients and was determined to be safe, but was not efficacious in these psychiatric disorders of schizophrenia and depression that it was studied. So in 2005, Sanofi discontinued development of the compound. What's interesting is that in looking at the mechanism of action, the NK3R is in fact located in the hypothalamus, and its relationship is with the GnRH neurons and the pituitary and the sex steroids. And so when you really look mechanistically of where NK3R antagonists may be relevant, you can see that this pathway, unlike its role in psychiatric disorders, this pathway seems to be very, very relevant. In fact, when you look at the knockout in human models on slide 29 of this pathway, you can see that GnRH, luteinizing hormone, follicular stimulating hormone, the sex steroids, estrogen, progesterone, androgens are all affected by a knockout of the NK3 receptor. And there are other drugs that are currently in development in the clinic that target NK3R, 2 in particular. One was developed by Molendo through phase 2, looking at vasomotor symptoms in menopausal women. That compound showed really great data, but in phase 2 was terminated for elevated liver transaminases. The second compound was a compound that was developed by a company called, I believe, Ogeda. That was a European company. They were acquired by Astellas in mid-2017, and they were also developing their NK3R antagonists to treat vasomotor symptoms in postmenopausal women. And you can see here on slide 31 data that they presented at ENDO in 2017, looking at hot flash frequency, and you can see that within week 4, a reduction of nearly 90% of the frequency of hot flashes sustained through the 12 weeks of the study, and when drug was removed, you can see that the symptoms returned to baseline. On slide 32, you can see that when the women actually had a hot flash, the severity of the hot flash was significantly diminished by upwards of 70% by the end of the study. So both the reduction in frequency and severity of symptoms by treatment of an NK3R antagonist is significant and sustained through therapy. Now, Astellas just reported phase 2b data in an abstract at ENDO 2 weeks ago, and they, in over 350 women, and they in fact found that there was, that all 4 primary endpoints were met in that trial as well. So significant efficacy, potential for, very good safety, certainly what was studied for osianimab in schizophrenia. What I will say is that postmenopausal women is not Acer's business model, but vasomotor symptoms in fact show up in a number of different rare and life-threatening disorders that we're interested in pursuing. So we're going to continue to take a look around at vasomotor symptoms in similar disorders that fit more of the Acer model. We've just ran an advisory panel with the University of Washington last week, looking at a variety of different indications and clinical development pathways, and we'll be providing additional detail on the development program for osianimab in the July timeframe. So the market opportunity for osianimab, again, we acquired worldwide rights to the compound earlier this year. There's some great clinical proof of concept studies with other NK3R antagonists that have demonstrated rapid and clinically meaningful improvement in vasomotor symptoms. They also looked at this in polycystic ovarian syndrome, so multiple disorders that are affected by this pathway. And again, I'll point out that Ogeda was acquired by Astellas in mid-2017 for up to 800 million euros at that time based on phase 2A data. The osianimab, specifically our compound, was shown to be safe and well tolerated. Doses studied in healthy subjects and schizophrenic patients when it was in development originally by Sanofi. We are refining the development pathway with top thought leaders, and again, we're going to be targeting these rare and life-threatening disorders. We'll be filing the IND in the second half of this year once that's fully elucidated. Slide 34, a quick overview of the financials. As I mentioned earlier, at the end of last year, we reported cash of $41.7 million, which is expected to be sufficient to get us into the first half of 2020. Our capitalization as of March 19th, we have a very clean structure, a little over 10 million shares outstanding. And when you factor in common stock, the options for the employees, that takes it up to just over 11 million shares. No warrants, no debt. And $87 million has been invested through August of last year, and top institutional investors are listed there. So in summary, Acer's pipeline includes, as I mentioned, 3 clinical stage programs, one that's pre-NDA and expected potential approval here in the next couple of months, and 2 additional compounds in development that we're pretty excited about. And really we have kind of a methodology, a platform, if you will, for evaluating these compounds. And certainly we're looking at additional compounds at some point to bring into the pipeline that kind of look very similar to these drugs, with a number of different milestones that are just around the corner. I'll stop there and see if there's any questions.